would like to introduce Mike Costanzo, the CEO and director of BreathTec. Dr. Costanzo enjoys a passion for science that has directed his career path firmly into the field of analytical chemistry. God help him. Over the past 6 years, he has investigated a variety of advanced experimental methodologies and instrumentation with the Yost Research Group at the University of Florida. This includes the use of mass spectrometry, novel ion mobility spectrometry, a lot of stuff. He obtained his Bachelor of Science in Chemistry at State University of New York in Buffalo. I grew up there. Before moving to UF. Upon graduation in 2015, he joined BreathTec biomedical team to propel his own bioanalytical research through the development of revolutionary clinical devices, utilizing his background in ion mobility, mass spectrometry, and exhaled breath analysis. BreathTec Biomedical was formed to propel innovative research in the area of point-of-care breath analysis as a medical screening tool. I'm going to let him tell you the rest of this story. So please join me in welcoming Mike Costanzo.
Thanks, Lee. I'm sorry I had so many words in there. There's a little more scientific of an introduction. And please forgive my voice if it cracks a little during this. That recent allergy spring has kind of taken a toll on my throat a little, but I'll make through. So I'm going to tell you a little bit about BreathTec Biomedical today. We're just going to kind of touch base on a little bit of the stuff we do, who we are, where we're going, and if you have any more questions, or any questions, I can answer them at the end, and then I'll even direct you to our website and that as well. And so, obviously, quick legal slide. Please don't steal our stuff, I guess. So quickly, I guess the very quick elevator pitch is BreathTec's goal is to breathe new life, so to speak, into diagnostic screening by giving a non-invasive alternative to the current methods that are used today. So a quick overview. BreathTec was formed, as Lee had said, to propel research in developing a commercially viable breath test. And so we can use this for early diagnosis, companion diagnostics, clinical compliance, as well as a few other things. And the primary avenue we're using to investigate our research is something called ion mobility spectrometry, more specifically a technique called field asymmetric ion mobility spectrometry, which I will give you a very brief touch on later in the slide. But to give you kind of a reference point for this, if you've ever gone to the airport and had your hands swabbed and then they test it for explosives, they're using ion mobility spectrometry to test it. And so we're using a very similar method, but instead of analyzing for explosives, we're analyzing for different biomarkers of disease in your breath. And so what this offers us is a potential for a low-cost, non-intrusive alternative to regular diagnostic screening, which could lead to notable advances in early detection in the general medical practice. Regular screening using a simple test would hopefully lead to significant advancement in remedial treatments that you normally do day to day. And this would improve the survival rates as well as quality of life of the everyday patient. And lastly, it would give greater access to diagnostics for a lot of the impoverished third world countries that might not have such. And so who's in charge here? Well, in terms of management, the top face should look a little familiar because I'm standing right here. So I am the Chief Executive Officer here at BreathTec, and I just got my PhD in analytical chemistry at the University of Florida, and we still work there today for now, and I'll talk about that in a sec. Our other management is actually all located in Canada, so we have Kale and Raj, our president, founder, and lead engineer, and they are a huge help in terms of getting BreathTec off the ground, and Raj himself is an MD PhD, so if you thought PhD was impressive, he's got both. And he's a surgeon currently working in Vancouver, and he lends a lot of ideas and help to further our research. In terms of the actual research, I work here in Florida with a few other people who are associated with BreathTec. So there's Jared Bach. He also recently graduated from the University of Florida from the same group as me in analytical chemistry, and he's the lead scientist at a company called Cannabix Technologies. They work very closely with us. We're working on a similar technology, just different applications. As the name would suggest, Cannabix, they're looking for THC in the breath, whereas we're looking for disease biomarkers. Guy Latore, who most of you probably know. He's a consultant for BreathTec. And then Dr. Richard Yost, he was actually my advisor at UF in chemistry. And he's kind of the scientific advisor for both companies. He's not employed by either company, but he just gives his advice when asked. And we currently work out of his lab at UF with the hope of eventually moving out. But right now, that is where most of our research is conducted, is at his lab at UF. So that's the who. Let's talk about the how. So fingerprints identify you. So if you ever watched the show CSI, they're always trying to find fingerprints, different markers to identify people based off a very specific code that correlates to them. Breath prints could hopefully reveal your health. What we do is look for a biomarker or compound in your breath that would be indicative of a specific disease. And the first question I usually get asked is, you can see stuff in your blood but in your breath? And the simple answer is yes. The more complicated answer is it kind of depends. I won't go too in detail for you, but what I did want to at least make clear was why we think we can do this. So many molecules in your blood can be found in your breath. The reason is there is a barrier in your lungs called pulmonary alveoli where molecules can diffuse just between your blood and breath pretty freely. Some molecules transition a little easier though. So some things are going to be at just absurdly low levels that are going to be really hard to detect, and so that's what's going to be, we're working on the technology to actually do that. Other things are just at such high levels that it's impossible not to see them in the breath. Some examples that you may have heard of before is if you can smell a very sweet tone on someone's breath, that's because they have a lot of acetones in their breath, and it could be a sign of ketoacidosis associated with diabetes. Similarly, if someone has liver disease and they're in the very late stages, you could smell kind of a putrid smell on their breath, just the different molecules that are coming off from these diseases. Now, the unfortunate part of your nose being able to smell it is usually that they're very far along in the disease at that point. So we want to be able to detect it much, much earlier than that. And so we aim for a non-invasive alternative to blood tests, urine tests, culture swabs, and we want to do this for various health-related concerns. Because again, each disease produces a very distinctive biological signature. Not necessarily a compound that's new, but you have, let's say, 10, 20 compounds, and their ratios are all going to change for each different disease. And so once you figure out a print for that disease, you can then use that in different people to identify a different disease. And so we're going to use our innovative technology to distinguish between these. So again, our goal is to provide you guys with a benchtop and or handheld breathalyzer. And so advantages of using breath analysis is that collection would be able to happen anywhere, anytime, once it's small enough. But unlike blood and urine tests, where you have to collect them in a very specific environment usually, maybe not urine tests, but blood tests and culture swabs, you have to be very careful. Breath tests isn't as, it's a lot more robust. You can kind of just collect it in a bag, collect it directly into the device, and it's not really gonna change too much, and it's not as much of a biological hazard as the others are. So there's a lot less minimum handling, or there's a lot less handling and storage procedures. With a lot of biological substances, you have very specific safety precautions that you need to follow. In breath, there are some, but not nearly as many, because you're talking about just air dissipating as opposed to spilling some contaminated blood or contaminated urine on you. And so that, again, allows for less handling and storage, a lot quicker results. We're talking about minutes as opposed to hours, days, or even in some cases weeks to get a culture back. And it's relatively inexpensive and simple to use. And so while our goal is a handheld breathalyzer, we're going to start with a benchtop analyzer. What this is going to allow us to do is validate the technology, but also validate and identify new markers for each of these diseases. And the bigger, more robust system will allow a broader range of diseases to be detected. And that's not to say that the benchtop analyzer isn't going to be portable in itself, because we're talking about something that's maybe the size of a desktop computer, maybe a couple computers, and so if that's somehow the smallest we can make it, which I'm very sure we can make it smaller, you can, if it's in a hospital, say, you can put that on a cart and wheel it around. It can move on it of its own. You don't have to set it down, and it's going to take up a bunch of space. So it's still relatively small, even though it's the bigger model. But the goal is still a handheld breathalyzer. And this would be a point-of-care device that, whether it's in a doctor's office, or even if we can make it small enough so that someone could take it home and use it with themselves at home. And this would allow real-time breath analysis of these different diseases. And so our key factors here are affordability, portability, and adaptability. So we have affordable electronics and sensors. These are all really small, disposable consumables. It's portable. As I mentioned, both the benchtop and handheld should be portable in their own in their own right, but the handheld's obviously going to be a bit more portable. And then it's adaptable. So two kind of things we're thinking of in the future here once this is developed is that you could have remote data capabilities. So if you do end up taking it home, when you test your own breath, that could wirelessly transport to the doctor's office where they can get your results. If the doctor's office happens to be collecting it and they're a little confused on what's happening, they can send those results wirelessly to another lab to help them figure it out a little more. The point being, we could have this remote data capability. And secondly, we could have software updates in terms of, I kind of hit on this a little bit, but essentially, once you print a disease, you have certain settings that you just put into this device. Each disease will have different settings. So once you figure out a new setting, all the devices should be able to be programmable to that disease. So let's say we only had 3 when we put out the first device that this hospital bought. And then a year later, we had 3 more. They don't have to buy a new device, they just have to update the software. And now they can read for those 3 new diseases. So that would be something that's very interesting. So what this all brings us to is kind of our goal is developing a disruptive technology. I'm sure most of you know what that term means, but in case you don't, it's essentially creating a new network, a new market by disrupting an existing one. And so blood, urine, culture testing, these are the norms at a hospital. And not even just a hospital, the doctor's office, when you're in the field, if you're taking samples from someone. And what we want to do is not necessarily completely get rid of them, because that's still the best way and most sensitive, most accurate way to identify something. But we want to build a market for breath analysis. The only reason there's never really been a market is that most physicians and clinicians don't know how effective breath monitoring could be, mainly because we've never had the technology to reliably and accurately measure molecules in breath. And we're hopefully providing that now, so that they can take that and kind of shift the paradigm to, instead of just going right to a breath test or right to a biopsy, right to a blood test, right to a biopsy, they can test the breath first. And so that's kind of combating 2 things. One, you're trying to detect it even earlier, because a lot of the time maybe you don't want to take a biopsy, maybe you don't want to test blood if you don't need to, because these are invasive things. And so we're hopefully, by giving a simple breath test that you can do every time you go to the doctor's office or just on a whim, you're keeping up to date on your health, and that way you're detecting things, potential problems that might happen sooner. The other side of that, a lot of tests that you conduct, or that physicians might conduct, come up false. You don't necessarily have that disease. But now you've had a biopsy, you've had an invasive procedure. In the case of melanoma, they'll cut that mole right off of you, and now you have a scar there. We also want to prevent unnecessary biopsies and unnecessary procedures. So we're going to hit both sides of that with this new technology in breath analysis. And so, like I said, I was going to give you a very brief overview of what the technology actually is. And so, as I mentioned, it's called Field Asymmetric Ion Mobility Spectrometry, or you can just call it FAMES. And what it does is essentially is just takes compounds, you ionize them, so you give them a charge, and then it separates them by something called their mobility. This is really just their size, shape, charge, and it does it in real time, and you can continually pump the sample into it, so you can breathe over and over again, and you can see every time a breath comes in. But the point is, it is essentially an ion filter, and so only molecules of interest are going to be allowed into this detector. And so the best analogy I could come up with is essentially a coin sorter. And so if you think of this, you're putting all your coins in, it's running through, you have different slots for each type of coin. And if you're doing regular ion mobility, all those slots are open, you're still separating everything, pennies, quarters, dimes, but you're seeing it all. And that's a lot to sort through still. What FAMES does is essentially closes off every single port except for, let's say, quarters. So you want to look at quarters, you close off all of them, and only quarters are coming through, so you can count exactly how many quarters there are. And then you decide, all right, I want to shift it over. I'm going to now look at dimes. So now you just count how many dimes are coming out. And this is kind of exactly what we're doing with disease markers. And so now, once we find what that marker is, we can tell the device, hey, look for this marker. It'll find it. We see if it's there. Move on to the next. Potentially, we could even have them all going simultaneously if something we're working on works out. But even if it doesn't, you can quickly and reliably change between these settings to just look for all these different markers. And so that's kind of the basis for FAMES. And so our target, again, is a handheld device. And right now, right on the market, it's not a breath device, it's a gas analyzer, but there's a company from the UK that produced a device that has this chip here. If you can see it, it's about the size of your pinky. And that is a FAMES device that's made on the micro scale. However, it's too small, and it doesn't really have the sensitivity, the specificity we need for stuff that you're going to find in your breath. To the left here, we have something about the size of your hand. That's something that was developed in Dr. Yost's lab, in my lab, before I got there. And that can separate everything, so you can see everything you want. However, it comes with a power supply that's like 70 pounds and is huge to carry around. So our goal as a company is to take these two and compromise. So we want to make it really small enough that you can make that power supply really tiny and portable, however, big enough to where we're not losing all the sensitivity that we wanted to begin with. And so, as I mentioned before, on the way to that, we're going to have a benchtop device, still not huge in itself, but that will initially validate our technology. And again, there's probably a market for that larger, more robust, more versatile device, whether in CLIA labs, clinics, hospitals. The main point being preliminary results we've done on our testing with our cell so far has been very positive. And so speaking of the cell, that's kind of where in terms of the business side of things, BreathTec is currently in the process of licensing a patent from UF. We're just finalizing the paperwork. And so that's for a FAMES device that Dr. Yost, my old advisor, had created. And so once we license that, that'll be the key kind of component of the device. And then we still have other efforts that we're analyzing as well and creating. So we need to be able to sample the breath. We need to be able to take those compounds from your breath and ionize them to be able to be analyzed. And then we need to detect them. And so these are all different technologies that we're developing in tandem that are all going to go into this device and work in conjunction with each other. In addition, we've been looking at a few other similar sensors. There's a lot of what are called electric noses or electronic noses out there. They don't give you the identification of the idea of what you're looking at. They can potentially sense a difference between one disease and not. And so we've been talking to a lot of those companies and seeing if they're willing to work with us to help us all kind of come together to perfect this technology. And so that was the why, or the how, I guess that would be. So this would be the what. So what are we actually going to look at? The device itself, if we're talking theoretically, can look at anything it wants to. I mean, you just got to tell it, all right, this is this marker, this is this disease, and it'll give you a print for that, and then you can use that over and over again. So I can't talk about every possibility, but I will talk about a few things that would be prudent to where we want to start initially. So when people think breath analysis, usually you'd think respiratory tract. So lung cancers and respiratory disease would be probably the first way to go, but a lot of companies are going after that. Dogs have even demonstrated the ability to sniff out cancer in breath, as well as just on the skin. And there have been numerous breath tests in terms of actual electronics, not including dogs, that have shown specificity between cancers, not just cancer, not cancer, but they can differentiate between lung cancer, breast cancer, other types of cancers. And this is good for us because some of this research even shows that breath testing is at least as accurate as x-rays, and that would allow for you to kind of not need to do as much of those procedures as I mentioned before. So instead of giving, for breast cancer, instead of getting a very uncomfortable mammogram, or for x-rays if you have a lot of radiation exposure, while these things are safe and comfortable enough that you don't mind doing them, if you don't need to, we can prevent that by hopefully giving you a breath test first and saying, yes, you might want to go further, or no, you probably don't need to do that. So early diagnosis is the key here with a lot of these diseases. Secondly, and one that I'm really interested in, is liver diseases and diabetes. So liver biopsies are invasive, painful, often the only way to go for a lot of these diseases, or extensive blood tests. But hopefully if we can characterize these diseases in breath, which we've already shown that acetone levels are pretty high, we know that glucose levels are higher, hopefully we can shift that over to let's look at breath first, and then if you need to, go into these more extensive tests. And even one of the hopes I mentioned earlier, if we can develop this point of care device that you can take home, maybe even eventually replace all these needles and syringes you're using to test your blood every day, several times a day, with a quick breath device. One other thing I had started mentioning earlier was other countries that might not have as much healthcare, so have diminished healthcare, especially compared to Americans. So while these devices that we're producing will be revolutionary to Americans, they will be even more so to these other countries that might not have as much access to that type of healthcare. Tuberculosis, namely, is responsible for more deaths worldwide than anything other than HIV. And so if we can develop a quick, reliable, inexpensive way to test for tuberculosis, as opposed to a trained professional flying out to this country and checking sputum samples under a microscope, which can take days, again, maybe even weeks, depending on the load they have, that would revolutionize medicine in those parts of the world. And so it would be instrumental in again, early detection of these diseases. And finally, in addition to diseases, I mentioned clinical compliance and companion diagnostics. So a costly issue with clinical trials is people not taking the meds they're supposed to. They say they do, but maybe the meds start to work and they think that they can just stop, or maybe they're not working and they're like, I'm not going to bother myself with this. And that's a huge risk when you're doing clinical trials, is people not actively taking their medication. And so this is a quick, non-invasive way to just make sure they're adhering to the regimen. And similarly, another major issue is efficacy. So making sure that, yeah, these patients are taking their drugs that they're supposed to be, but also that the treatment they're being provided is actually effective. We can monitor the breath for degradation products, biological products, and make sure, yes, this treatment is actually working, and they're taking things as recommended. So we can ensure that the therapy is working as designed, and all of the patients are complying with it. And so lastly, I just wanted to mention, we are working a lot out of the Yoast Research Group currently, and so that's over in the chemistry division of the University of Florida. And the good thing about working there right now is that a lot of the research that comes out of that group is focused on instrumentation, fundamentals, and application, and that is the three main things we're doing with our company right now. We need to build this instrumentation, but we need to make sure that the fundamentals are there, that we can prove to the physicians and the clinicians in that what we're doing is sound, because again, this is going to be a paradigm shift, hopefully. And then we need to talk about different applications that we're using it for, different diseases, different compliance issues. And the research in this group does amount to a lot of data that goes out into the world. And so I was a part of this group when I was a grad student, but I did want to mention that we are working there now, and then we'll be moving out in time to our own lab space as we move further along in our research. And so with that, I'll take any questions, and then if you have any other questions after the talk or just in general, you can always visit our website, which is up and running at breathtechbiomedical.com. So thank you.
You got a lot on your plate.
I'm curious on the breath side of the measurement, if you will. In blood you measure parts per million, so in breath you've got to measure the volume of air as well. So two-part question. The first would be is, how sophisticated does that have to be for your device to work? The other question, it seems that air density would have an effect on this. If I lived in Death Valley versus Santa Fe, the molecules of anything, even oxygen per cubic measure, are going to vary. So how do you account for that?
Good question. Right now, that's actually something I mentioned when I said we still have to figure out the breath sampling portion. I kind of lumped them all together. That is a huge factor that we need to overcome at this point. We're currently doing studies to figure out what that correlation is, first between blood and breath of specific molecules, and then how can we accurately and reproducibly see those molecules in breath, not only from person to person, but just from that same person's breath to breath, because they vary. And so we've been tooling with the idea of a possible internal standard that we can use, whether it's something we spike in or if it's just something that's, as we progress in clinical trials and whatnot, if there's some molecule or some molecules that seem to be constant in your breath, and we've seen a few so far, you can use them as an internal standard to compare everything to. And so even if there is fluctuation, you look at that kind of comparison molecule and say, this is here, so we know to account for that. And so that's kind of the way that we'd have to do it, is have some sort of standard to reference against.
Hi. So this is probably going to have to be an FDA-approved device for clinical use. How does that work with when you talk about software add-ons or upgrades for additional tests? Is this going to be like a platform, and the FDA is going to kind of generically okay it? Are you going to have to recertify every time you add a new test?
I actually don't know the answer to the question right now, but those are things I've thought about. I am hoping and assuming that the initial device that's going to be the hardest to push through the FDA. We're using, I believe it's called the predicated method, in that we're comparing it to what's called mass spectrometry, which is kind of like a standard nowadays. Right now we actually have it on the front of a mass spectrometer. Eventually we're going to take it off and have just its own detector. But because of that, we're going to use that to say, look, you trust mass spectrometers, that's what we're using to detect it, we're going to validate it with that, but now it's its own device. And we're using it as a screening tool as opposed to you definitely have this disease type of thing. So that'll ease that regulation a little bit. Still regulations you got to go through. But it is a good question about each disease. I imagine each one might be its own kind of push to go through, but if they're similar enough, they might say, okay, well, it's very similar to that. So that's something I haven't.
Mike, I can help you answer.
Oh, okay.
It's,
Wait.
Oh, yeah, you need a microphone.
Mike, I can help you answer. So essentially what we would do is do what's called a biomarker discovery submission. So we're going to be identifying biomarkers. Some of those biomarkers have already been discovered. In those cases, we're going to have to cross-license the patents associated with those biomarkers. Some biomarkers are going to be new IP. But in any case, with regards to the FDA, every time you submit a series of biomarkers, you have to validate those biomarkers using existing technology. So you do a biomarker discovery submission, essentially. And that'll have to be done with every disease, every biomarker, unless it's already been validated and approved by the FDA, because we're using a 510K process in a predicate device. But yeah, it's going to be a lot of regulatory work involved.
Along those lines, have you already determined some of those biomarkers? You already know that they're patented and that you're going to have to approach that company to get licensed?
There are not so much companies as I've read in the literature. There are a lot of biomarkers people have pointed out that might not be validated in terms of the FDA, so we probably have to revalidate them. But there are some that we've been looking at that are patented that we'd look at licensing.
So we're very early in the biomarker
Yeah,
discovery.
we're kind of going down every avenue at this point.
So my other question is, why would somebody need to have this handheld device if they already know that they've got the disease?
For the most part, I'd say it's going to be used to initially find that disease. But the one thing I had mentioned was for diabetes specifically. So yeah, everyone knows they have diabetes if they have it, but they have to prick their finger several times a day to test their blood glucose levels. So I might not have mentioned that part, is it's not always just diseases, but you can go specific molecules as well. And so if you can monitor glucose on your breath, then instead of pricking your finger constantly, you'd have this little device that you could measure your blood glucose with, or some other variation of that for some other disease. That was just the first kind of example I would think of. So it depends on what you want to look at, but if you can train the device to look for it, then you can use it that way, if that makes sense. Not to be super vague, but...
Well, there's a lot of activity right now in this specific area. So who is your biggest competitor, and why will you advance beyond them?
In terms of biggest, I don't know about the largest, because it feels like it's still there's not a lot being sold, there's a lot being patented. And a lot of them are the same. Not to be like, oh, everyone's is all the same except for ours, but every company I've talked to and we've visited and whatnot, there's one over in Israel, Technion, there's one in Pittsburgh, there's the one in the UK that I mentioned, there's a couple in Florida. They all do what is essentially an electronic nose, electronic sensing. And what they'll do is take a diseased state breath, so someone who has this disease, they'll breathe into it and they'll collect the data on the sensors, and then they'll have someone who's healthy breathe into it, collect that data, and run it through whether it's a patented algorithm or just some sort of statistical analysis, and it determines the difference between them. So they don't actually know what they're looking at. It's just, hey, we have a difference between these diseases, buy our product. And that's perfectly fine if that's what you want to do, but convincing people that it works is the hard part. And so what we can do is take our device, which filters everything out, and yeah, is essentially also a fingerprint device, but it works on the front of what's called a mass spectrometer that we've mentioned a couple times, and that specifically identifies compounds. And so once you identify all the peaks, all the different things you're seeing, you can then unhook it from the mass spec and say, these peaks are validated, we know exactly what we're looking at. So the main thing we have over a lot of the competition, if not all the competition as far as I can see, is that we can actually validate and identify what we're seeing, as opposed to just validating there is a difference or not. It's, here's that difference. And then what you could even use from that, and something that I always found interesting, was if you can find new biomarkers or new compounds that are coming from these diseases, you can even better understand the pathology of these diseases. So we're even offering, like, maybe if you're not using our device at that point, you identified it with our device, and then you're like, let's do a bunch of research on this compound now, and we can actually better understand the pathology of a disease, that would lead to better treatment, better better ways to combat that disease.
Are you working with NIH as well?
Personally,
This would be an area that they would be really anxious to fund.
we are in the process of writing STTR, SBIR grants, and that was the company that the one main group that I was assuming would probably be the most interested in it. The Yoast group specifically, like I've had NIH grants. The company right now specifically doesn't have an NIH grant, but that is a company that we'd go toward because they are obviously very interested in that type of research.
Yeah, question. Do you have to condense the breath onto the surface of your sensor? Like in other words, do you have to, when you breathe into it, does the breath need to condense down to some concentration in order for you to be able to have enough sensitivity and specificity to detect what you're looking for?
As of now, no. We really want to stay away from that. So the most common way to analyze breath right now is something called exhaled breath condensate. Whereas you breathe it out, you're condensing it into a liquid form, and then you inject that liquid into some instrument, whatever it is. The problem is, in addition to all the molecules, which are already at a really low concentration, you're condensing a bunch of water vapor, because most of it's water. And you're diluting it in pretty large factors. You can always dry it down. Well, I don't want to go into too much specifics, but you can work around that, but it's a big problem. So what we've been doing is breathing directly into the device because we have an ionization source that can analyze gases. And so it's just in the gas form. And so we're not changing it at all. We're going to see if that's sensitive enough to work. If not, there are other things before we have to go down to condensing it, but we're hoping to keep it in the gas form. Whether you collect it ahead of time or breathe right into the instrument, we want to keep it in the native form that it's in, so we don't have to worry about any changes.
Hi. Very good presentation. My question is, in terms of the research landscape, Lee just asked about the grants, and you're obviously working very, very closely with the Yoast group, where are the other research nodes and universities or other medical institutes? Is there a lot of activity in this area on the basic research side?
With our technique specifically, you mean, or?
Yeah, well, or just the whole notion of identifying biomarkers in breath.
There's definitely a lot of people identifying biomarkers in breath. I know Banyan biomarkers used to just be biomarkers. I don't know if it was specifically in breath. But a lot of the literature I'll look into is just worldwide. Is a huge a huge look into breath biomarkers in Europe specifically, and a little in Israel and that. So it is more in over there as opposed to here, but there are a few little pockets of breath analysis people, but no one that I know of, with the exception of one other company in the UK, is specifically using FAMES and breath analysis. So we were the first group to do that because we were working with that company in the UK, and we showed them that, hey, you could probably look at diseases, and they kind of took it and spun it off on their own too. So, but as far as I know, there's only 2 doing those specifically, but there's quite a big group doing breath itself.
Any more questions? Thank you, Mike.
Thanks for having me.